This week in lab, you're going to be exploring photosynthesis and cellular respiration using algae balls. Right, that might sound a little strange to you. Let me explain. So remember what you've learned about photosynthesis. If you haven't studied very much about photosynthesis yet, stop the video now and go study photosynthesis. All right, think about why photosynthesis is important, if photosynthesis is enough for a photosynthetic organism, or if they need to do something else with the products of photosynthesis to make them useful to the organism. Right, remembering from our sort of basic adorable little video, using carbon dioxide and sunlight and water, there's the water, we're going to be making sugar. Right, the glucose thing is wrong. And the byproduct or waste product is oxygen. Follow along with your lab handout for this, but I want you to think about some specific questions to get you ready. Remember, this is about learning. Don't rush through this in order to get the grade. Think about this. People would say chew on this. You really want to understand it because photosynthesis is the reason why the world exists. It really is the way that it is. So you want to think about, where do organisms get their energy? Not just you, all organisms. What's the ultimate source of energy for the biosphere, the entire world, for the most part? I want to think about how this happens. How do we go from, let me find the right picture, this tiny, tiny little seed. And we can see the seed in relation to a penny. How do we go from that to, let's find the best picture here in case you've not seen a redwood. Something like this. Right, these are huge. This is a giant sequoia or a redwood tree. You can get these and grow them in your yard. There's one called the Dawn redwood that can grow in these conditions. These can be enormous. How do you get from a tiny, tiny seed to this enormous tree? Where does the plant get the materials to build such an enormous structure? Where do trees get their mass? That's the central question here.
Trees are some of the biggest organisms on the planet. But where do they get that matter to grow?
Which nutrients out the ground? Just out of soil, really, yeah.
Goodness out of the soil, I suppose.
Comes out of the soil.
Yeah.
Goodness.
Goodness.
Why isn't there a big hole around the tree where it's taken out all the soil?
So does it say gradually that the soil has time to recover?
Now, I think it's intuitive to believe that the tree gets most of its mass from the soil, because you can see those roots digging into the soil and they must be taking something out of there. And I mean, a tree looks like dirt and it feels solid like dirt, but it's not. In the early 1600s, a scientist named Johann Baptist van Helmont tried to figure out where the mass of a tree was coming from. So he got a pot of soil and very carefully measured the amount of soil in there. Then he planted the tree and took care of it for 5 years, making sure that no soil left or was added to his pot. And at the end of this experiment, he weighed the tree to find that it was 72 kilograms, but the mass of soil had only decreased by about 60 grams. This was pretty strong evidence that the mass of the tree does not come from the soil.
I've never thought about that, actually, because they don't really eat anything, trees.
They don't eat anything?
No.
They don't eat anything?
Water is all they absorb.
That's all they eat?
Yeah.
They don't eat anything else?
No.
That's all they eat?
Well, presumably from the water and the nutrients from the soil.
Is there anything else that you need besides the soil and the water?
I suppose that's all you need, isn't it?
To make wood.
Other than the original seed for that particular tree, I
The
suppose.
seed and the soil and the water, and that makes this big tree.
Of course, Johann Baptist van Helmont did conclude that the tree was made entirely of water. Now, while that's not correct, at least he was on the right track, realizing that the matter of a tree doesn't come out of the soil.
The sun, energy, yeah.
The sun energy?
Yeah.
Are they converting energy into mass, or Do you know what I mean?
Yeah, I don't
Like
know.
there wasn't stuff and then there was. Like where did that stuff come from?
Magic.
I have no idea. My question is, where do they get that mass to grow big?
Sun. Get it from the rain and the sun, presumably.
Light. Sunlight.
And the sunshine.
The sunshine? Does the sunshine add mass to the tree?
Well, yes, it wouldn't. They wouldn't grow without it. I don't know whether it adds mass, but they wouldn't grow without it.
Of course, the sun's energy is needed for the tree to build the matter into its branches and leaves. But the sun itself, the energy, is not matter.
Well, I suppose we're going to put air into this as well. If
Yeah.
there's got to be something out of the,
What is
a
it about
gas,
the air?
a gas in the air that it needs as well.
Oxygen.
The trees need the oxygen.
Yeah, need the oxygen, don't they?
And I guess oxygen.
The oxygen, of course.
The oxygen? Are there any ingredients that we're missing?
Carbon dioxide.
Carbon dioxide.
Carbon dioxide.
So would it surprise you to find out that 95% of a tree is actually
Carbon dioxide.
from carbon dioxide? Trees are
much,
largely
yeah.
made up of air.
Surprising. Yeah.
I need this reaction like, oh,
Holy shit.
So as it turns out, trees are mostly made out of air, out of the carbon dioxide that they take in. And what's interesting is that we breathe out carbon dioxide and water. That's how we lose mass. But it's the exact same substances that trees breathe in to gain mass. So if you can imagine a closed system where it was just you and a tree, you would breathe out that carbon dioxide and water, the tree would take it in, so you would get smaller while the tree is getting bigger. And in a sense, you're becoming the tree.
So think about that. Is that true? What's going on there? There'll be some quiz questions throughout that, so don't worry, you will understand it. I want you to really think about it. Most of us did not get a good introduction to photosynthesis in high school, and because of that, a lot of us have developed a mental block. Take the block out, pop it out, get past it. Because this is probably, I would argue, one of the most, if not the most important processes on Earth. I want you to think about what is algae. Are you going to be using a specific type of algae today called chlorella, which I always think chlorella de Vil. So if you've seen 101 Dalmatians or read the book, that makes sense to you. If not, go read 101 Dalmatians. So chlorella is a protist algae. You may also be using a mixed algae culture today. Let's look at some algae. So these little dudes are chlorella, the round ones. You should be using a high concentration of chlorella in the solution. I've been growing them on my porch at home under some lights. I'm sure the neighbors think that I'm growing something else, but I'm not. What process does algae use to transform energy? Does it use just one process, or does it use more than one process? You're going to be exploring that today. What I want you to think about is photosynthesis is the process that organisms use to transform energy from sunlight into a usable form of energy in food. Right, so a photosynthetic organism uses sunlight to drive the process of making sugar, right, and then it's going to use it. So if you go through all this work to make yourself dinner and you put your dinner on your plate on your table, are you done? No, not at all. Right? You need to eat it. So organisms need to pair photosynthesis with cellular respiration, making the food in photosynthesis, using the food to generate ATP to run the cell in cellular respiration. These two processes need slightly different conditions, and if one is higher than the other, you're going to see different results in your data today than if the other is higher. All right, so go to exercise 1 in your handout and fill out those questions. What color do you think will result in the highest rate of photosynthesis and why? What would you expect with a higher rate of photosynthesis? An increase in pH or a decrease in pH? Do you think the tubes with no light will have a higher or lower pH compared to the tubes without a color filter and why? What do you think is going on when there is no light? Think about that. What's going on? Was it photosynthesis or cellular respiration and why? And will the pH in the tubes with no light change at all? Why or why not? So pause the video here and answer those questions. I'm not going to answer them, so don't think, oh, if I keep going, she'll answer the questions for me. I'm not gonna. I want you to think. All right, you're going to be using algae balls today, and these are going to be in little test tubes. Basically, here's a picture of a tube of algae balls. I'm going to have a short tutorial on how this works. Plus, the music's fun. I was spelled wrong. I didn't make this. There's our little algae balls. So you're going to put your algae balls into little tubes or cuvettes, depending on what we do with it today. And then you'll put your algae balls under a specific color of light or under no light at all. You want to think about what it would be a good positive control and what would be a good negative control and what those will mean. You're going to be measuring pH as an indicator of what's going on. So what we have here, I'm not going to even bother to show the video because it's really not that informative. All we need are these equations. So if we have photosynthesis, which you're showing us here, right? We have CO2, we have 6H2O, we're going to have sugar and oxygen produced. And if we have photosynthesis, we're going to have one result happening. If we have respiration or cellular respiration, we take the sugar, we add oxygen, we get CO2, and then we get water. Right, CO2 in water is going to be converted to carbonic acid. So the more acidic the solution is, the more CO2 is in it. As CO2 is used up in photosynthesis, right, that carbonic acid is broken down, the carbon dioxide is fed into photosynthesis. So that means that we're going to lower our acidity, which means we increase in pH. So if we increase in pH, that means that we are having photosynthesis going on. If we decrease in pH, that means that we're having cellular respiration going on. Now keep in mind these processes are happening together, right? So you want to think about who's winning, right? Who's winning, right? They're both happening. Who's winning and why? You're going to be using a color scale from Biorad to measure the pH. So there'll be an actual color change in your solution, which is really kind of cool, right? You're either going to compare it to the scale, you'll hold your tube up to the scale, or we will use a spectrophotometer, depending on what's available. So what do you do now? All right, you want to look at your protocol, and we'll look at it together. So you want to turn the page to the protocol or the procedure. At the end of today, you will have the following data. You'll have pH in relation to wavelength or color of light, in the presence or absence of light, and in relation to time. Remember to wear goggles when handling any liquids. We have a 50 ml tube that we're shown here. We have transfer pipettes, and we have a beaker, which I'll show in a minute. You'll have a 50 ml blue capped tube with 6 mils of 2% sodium alginate already in it. This stuff is really thick, so I will have prepared that for you. You'll add 6 milliliters of an algae culture to this tube and gently invert it. Invert. Gently to mix it, transfer it. Now when you get your algae, you want to try to suck up a nice thick culture of algae. So if the tubes have settled, that's good. Right? Take your pipette, go down to the bottom of the algae container and suck up some concentrated algae solution. You want nice dark green balls. You're going to pour 100 mils of calcium chloride solution into a 250 ml glass beaker. There's the beaker. All right, and then you're going to drop the algae into the beaker. To do that, you take a 20 ml syringe, right? So here's the syringe, we've taken the plunger out. You're going to hold it over the beaker of calcium chloride and pour the algae mixture into the syringe. And then you're going to slowly move the syringe around. It looks like this. No.
I saw that. Oh, I hope she needs this video. Is this a video? Oh.
Okay, so that was a video from the fall of 2019 semester when they did this experiment. I left the sound on just because it's adorable. So that's actually what you will see. Usually see algae balls start to form. They look a lot like caviar. This same technique is used in culinary or restaurants or however you want to talk about it, fancy chefs. You can actually take lots of different foods or juices or drinks and you can turn them into something that looks like caviar. It's a really cool fad. People actually even do it with mixed drinks. If you're over 21, you can try that. If you're not over 21, you can do it with fruit juice. But make sure you're using food grade stuff. All right, once you have dripped all the algae in, you're going to let your balls sit for 10 minutes and they're going to set up. Right? At first they're going to be too squishy. And this would be a really good time to take a ball out, squish it on a microscope slide, and look at what the algae look like. This will give you an idea as to how concentrated your algae is in your algae ball, and that'll be important for when you write up your results. So while you're waiting, maybe take out an algae ball, stick it on a microscope slide, squish it with a cover slip gently, and look at it with a microscope. All right, while you're waiting, label your tubes. Label 2 of each of the following. White light, so you need 2 white light. You need 2 no light. Okay, think about this. Which one's your positive control? Which one's your negative control? And then you label 2 of your color number 1 that you are assigned and 2 of your color number 2 that you are assigned. And you're going to have 2 that are no algae, right? Because you want to see if just the solution that you stick in this tube causes the stuff to change color, right? If it changes color on its own, you need to know that. Right, after your 10 minutes of letting your algae balls sit, you're going to dump your algae balls into a sieve or a colander, very fine colander, right? Don't use like the big holes colander. You can dump it into a sieve or you can dump it into a coffee filter over the sink, and you're going to rinse your balls with deionized water in a squirt bottle. I would use a scoopula to transfer your balls into the test tubes. So use a scoopula to transfer about 20 algae balls to each of your tubes because you want it to be equivalent. Fill each of these test tubes with deionized water, incubate them for 3 minutes because you're washing them, and then you're going to carefully remove the water with a plastic pipette. I want you to then add 15 drops of your indicator solution, cap your test tubes tightly, and gently invert. Right, then immediately, this is time 0, right? This is at the beginning of your experiment. Immediately use the pH scale to determine the pH of each tube and record it in a table like the one shown at the right, similar to the one shown at the right. Doesn't have to be perfect. I think we're missing the no algae on there just because it wouldn't fit. And now apply your treatment. Right, wrap it in foil if it's got to be in the dark, or put it in a dark box, or put it in a drawer. Put it under white light, the same type of white light that you're using for the color, the same type, the same intensity. Right? Don't put it in a different type of light. Put it under color 1 and under color 2. All right, every 10 minutes, you want to do this quickly, right? Because you have to take it out of the light that it's in or the condition it's in to check the pH. Do it quickly, because otherwise, if you take it out of its no light treatment into the light to test the pH and you wait too long, you're not going to have an accurate reading. Right, we're going to do this for 40 minutes. You look at the end, you'll get the total change in pH, and you really want to pay attention to whether it was an increase or a decrease in pH and what was happening over time. Right, record your data. We're going to share our data in the class, and then you will graph this.